thank you for joining this webinar. My name is Maaike Everts, and I'm excited to host today's description about the TRxA Bridge Awards. I'm joined by my colleagues, Mark, Kyla, and Michelle. So we have the next hour to talk about our RFP and also have some time for question and answers. Just a logistical housekeeping note, if you do have a question for us, feel free to put it in the Q&A box. There is also a chat box at the bottom of your screen, most likely, but really for question and answers, use the Q&A box as the name implies. So with that, let's get going. Wanted to start by introducing us as the team. So again, my name is Maaike Everts. I'm the Executive Director of the Translational Therapeutics Accelerator. I've been with Critical Path Institute almost 3 years now. Spent most of my career at the University of Alabama at Birmingham as a professor and have a background in pharmaceutical science. So Mark, maybe you can introduce yourself.
Yeah, my name's Mark Drew. I'm the Director of Drug Discovery and Development at TRxA. I'm a medicinal chemist, spent most of my career in the pharma industry.
Great, thank you. And then we have Michelle.
Hello, everyone, and welcome. I am Michelle Morgan. I'm Associate Director of the Translational Therapeutics Accelerator. I've been with CPATH just over 7 years and look forward to sharing information with you today.
Great, and finally we have Kyla.
Hello, everyone, welcome. My name is Kyla Edding. I'm Project Coordinator for TRxA, and I work with Michelle on the operational side of things, and I've been lucky to be with the team for over 2 years now.
Fantastic. So we will have the rest of the team go off camera and mute, but they will be in the background answering some of your questions via the Q&A. And of course, like I said earlier, towards the end, we also have time to answer some of your questions live so that more people can benefit from the answers that we may be able to provide. So let's start with our mission. TRxA is a global drug discovery and development program that supports academic scientists to advance new cutting-edge therapeutics from the lab to the clinic. So you see a few words underlined and highlighted here on the slide and wanted to briefly touch on each of those. The first is the word global. We don't have geographical boundaries or restrictions to who we can fund. So anywhere in the world, wherever you live, we are happy to consider your application. And we just try to find the best science, no matter where it comes from. The second set of words is academic scientists. This also includes nonprofit or research institute scientists. We're looking for projects that are still in a nonprofit environment. So this also means that we cannot fund for-profit companies. So if you are here and you're owner of a small company and you're looking for support, unfortunately, that's out of scope. But we are happy to support anybody who still works in an academic institution or a nonprofit research institute to get you ready for that commercialization of your program. And then finally, the word therapeutics. We do not support diagnostics or devices or anything like that. We're really here to support drugs. So in summary, we are looking for therapeutic molecules in lead optimization and IND enabling studies to support, and we'll talk a little bit more about details later in the webinar. For those projects, it is important that composition of matter, intellectual property, is either in place or obtainable. That also means that method of use type of patents, for example, for repurposing, are also out of scope at this point in time. And then finally, the intellectual property still needs to be within the institution, within the not-for-profit entity, whether again it's the research institute or a university. It cannot have been optioned or out-licensed to a commercial entity like a startup company or an existing biotech or pharmaceutical company. So why do we exist? Let's talk about the value of death for a moment. Of course, many of you on this call are aware what this entails. But when you look at this graph, we wanted to illustrate it graphically. On the left, academia and other nonprofit research institutions really excel in a lot of basic science, and there's a lot of resources from federal governments that support this kind of endeavors. On the right of the slide, you see industry, and this is pharmaceutical industry and biotech. They tend to really focus on supporting projects when they're in the clinical stage, especially late phase clinical trials. It starts to become very expensive at this point in time, and they have the pockets in order to execute those kind of that kind of work. So this middle part is called the value of death, and it's really about translational science. So there's a couple of aspects of translational science that make it that complicated. They're listed on the bottom. So regulatory considerations, reproducibility issues sometimes get in the way of companies investing into academic technologies. It's risky. A lot of drug discovery projects don't work. So the earlier you are in the process, the higher the risk you take as an investor into hopefully getting a return on your investment later down the line. Collaboration issues, you need to have multidisciplinary teams and so on. So lots of different aspects play into this valley of death. And there's a wonderful paper by Chris Austin, who used to head the NCATS Institute at the National Institutes of Health. I highly recommend you taking a look at that. Opportunities and challenges in translational science. So this is a paper from a few years ago, and I don't expect you to be able to read all these small numbers on letters on the left, but it kind of shows all the different colored boxes are different aspects and disciplines in the ecosystem that all need to come together in drug discovery and development. So basic science research and target identification is very important. Of course, that's where all the new ideas come from. But then also target pharmacology and biomarker development, that's highlighted here by this gray box. Lead identification and then optimization, candidate selection, IND-enabling studies, and scale-up for manufacturing. That's what we typically think about when we think about drug discovery and development. That's highlighted here on the right with this first green box and then this blue box, which is a little bit more linear. But again, lots of arrows are going to other colored boxes as well, like clinical research and development, regulatory review, post-marketing, and medical landscape in general. So again, highly recommend you take a look at this paper, but it shows that you really need to be almost multilingual in order to operate in this ecosystem and be successful. So accelerators like TRxA can reduce risk, and programs like ours, but we're not the only ones in this system, of course, but we try to force an organizational culture of collaboration. We build together interdisciplinary project teams where different people with different backgrounds come together to provide input and expertise into the program to make sure that it advances towards the clinic as quickly as possible. So for example, in order to get funded by an accelerator like ours, there's a lot of target scrutiny. So what is the molecule in the body that your drug in development is supposed to act on? Is it druggable? What's the role of the target in disease? Has it been clearly defined? What are the chances that it's actually important? What's the data around this target and so on? And then also when you have these interdisciplinary teams, we provide input on how to design your assays if you don't already have a strong idea on how to do that, but also to use what's called orthogonal assays, which are assays that are measuring the same thing but in a different way. And it really eliminates the chance of false positives or false negatives just due to the way that you measure something. Then there's comprehensive compound characterization and development input. Again, many times in academic groups, there's not really an awareness of all the things that you need to evaluate on a compound to make sure that it's a good drug or drug to be. And when you have this multidisciplinary input and guidance and mentorship, there will be some guidance, some input into like, hey, we need to check microsome stability or Ames or cytochrome P450 inhibition, all those kind of things that are typically a little bit more further away from a usual biology-driven lab. Then there's usually a large emphasis on evaluation of target engagement. So the drug that you're making, does it really actually interact with your target? Did you measure it in vitro, but also in vivo? What's the selectivity of that? And do you have the right proof of concept to then move forward? And then we really encourage early engagement with pharma and biotech, venture capital as well, to get feedback on your project. And if you talk to a couple of different parties that may ultimately be interested in licensing your asset, if you get the same question from all of them, you probably need to answer that question, right? So it really helps you identify what is that blank space in your data package that you really need to have an answer for before somebody gets interested enough to maybe invest in it. And then we personally also require regulatory science input. We have a lot of colleagues within the Critical Path Institute that have experience in that as well. But even before that, we really ask all our applicants to establish a TPP or target product profile. And it's a document very often used and commonly used in pharma. But again, not that typical in a more basic science type lab or any academic lab for that matter. And it's really a document, it's a living document that highlights what is your target population, what's the desired route of administration, what is the desired level of effect that would make a meaningful impact on how somebody feels, functions, or survives. What is the level of side effects that might be acceptable. And the more you learn about your program as you develop it, the more defined the target product profile becomes. But even when you're at the start of an exercise, doing this target product profile will kind of make you think about a couple of things like, oh, what is the route of administration that will be acceptable to this patient population? I mean, I was thinking of an oral, but maybe sub-Q is fine, right? Or the other way around, I was thinking intravenous would be just fine, but if our competitors all have oral medication, maybe that's not going to be, acceptable by the patient population. Just having those conversations and thoughts about this, maybe talk to a clinician who treats that patient population if you're not a clinician yourself. It really forces some thinking around what's the ultimate use of your drug that you're trying to develop. So here we are, the Translational Therapeutics Accelerator Bridge Awards. So we try to support the next generation of therapeutics. When you look at the timeline on the bottom, so you start with early discovery and then you go all the way through the clinical trials to phase 4 and beyond. Where we operate, where we really try to make a dent into supporting your projects to get further along this pipeline, is lead optimization and IND enabling studies. This is really where this valley of death is pretty tricky to navigate. Basic discovery, there's again quite a few federal programs, your typical in the US at least R01 type funding that's available. Phase 2, phase 3 in particular, that's typically where larger pharmaceutical companies start to step in. But this in-between phase is a little hard to get money. And exactly what do I need to do, right? So that's where we can help. So this is in summary, everything I just talked about. The problem is the transition between academia and industry, this valley of death. There may be a lack of understanding of what kind of data package do you even need to put together to get it to be of interest to industry partners. And then also these regulatory aspects of drug development. It's not something you typically think about when your main day job is to think about new biology or, treating patients and so on. And then sometimes academic studies lack reproducibility. So we try to address that as well by getting third parties to repeat your key experiment, again, to reduce risk. So the solution for us is to provide both funding and expertise. So the expertise comes in the form of regulatory guidance, and we assist in study design and implementation. Really, we're an extra set of brains that help you get your project to move forward. And then we also support the use of contract research organizations to maybe validate your findings. Again, if you have a neutral third party that has the same result of a key experiment as you do, a potential investor is going to have much more confidence that this is something real. And then also we like using CROs to execute parts of the work. Many of the assays, for example, around pharmacokinetics or safety panel screening or microsomal stability, it's done much cheaper, faster, and better at contract research organizations rather than figuring out how to set this up in your own lab. So we're a pretty unique model. We're an independent nonprofit organization. We're really a neutral facilitator. We do not want to own any intellectual property. We don't claim any IP rights, so that all stays with you. And we really can leverage the Critical Path Institute's regulatory knowledge and relationships that we have. And I'll talk about that a little bit a few slides from now, what CPath in general actually does and how come we have such regulatory expertise. And then we provide project management to facilitate the planning and execution of the project. And really this comes in the shape of us having monthly meetings. There are fun meetings where we talk about, so what happened last month? What should we do next month? And again, it's science driven. It's not a, oh, what did you do for me lately? But it's really a collaborative, fun way to get together on a regular basis to make sure that the project is heading in the right direction. So we are funded ourselves by the Frederick Gardner Cottrell Foundation. And this was established by Research Corporation Technologies, which is a venture capital group in Tucson, Arizona. So they established this foundation a lot of time ago, December 1998, and they provide financial support for scientific research and educational programs at nonprofits. Critical Path Institute is a nonprofit, so we were very fortunate to receive a large grant from them a couple of years ago to start this TRxA program. So a little bit more about Critical Path Institute as a whole. So we host a lot of public-private partnerships. And these public-private partnerships foster development of new evaluation tools to inform medical product development and regulatory decision-making. So the way we do that is we convene scientific consortia that have industry partners, academic groups, government agencies, and so on, also the regulators, and they come together to share data and expertise. So in these groups, there's active consensus building around what's a problem that is really good to tackle in a certain therapeutic area, for example. Let's share the risk and the cost to tackle that problem, bring the best science and the broadest expertise to the table, and come up with an action plan to figure out how we can solve a problem. Now, in all of this, there's iterative participation of regulatory agencies. So it's EMA, FDA, PMDA, and others around the world. So as part of these consortia, there's regular calls. The regulators are part of these calls. And together, again, there's development of a new method to, for example, assess the safety and efficacy of medical products. Now, throughout that process, the goal is to get official regulatory endorsement of these novel methodologies and drug development tools. There's a couple of examples of what these tools could entail. It could be a biomarker. So we have a consortium called the Predictive Safety Testing Consortium, where they're looking at kidney injury biomarkers, both preclinical and in early clinical stages, to see, can we predict what kind of drugs may end up having a kidney injury problem? There's a similar, in that same group, there's an effort in complex in vitro models to predict liver injury. We also have groups in therapeutic areas, for example, neurodegeneration, looking at certain imaging methods to stage patients that can be used for clinical trial design. It could be the development of clinical outcome assessments, like questionnaires for parents of kids with asthma. There's a lot of science that goes into those drug development tools, and Critical Path Institute is really good and strong in that science. So how do we then, as an accelerator, operate within the CPATH context? So again, CPATH is this neutral, pre-competitive ground where we have industry partners, academic groups, regulatory partners, patient advocacy groups. The voice of the patient is very important in all these deliberations, and in other government agencies. So we synergize with these more than 20 consortia that we have. And again, the consortia are focused on drug development tools and their regulatory endorsement. And we directly focus on developing drugs, hopefully the ones that are being developed by you in your lab. But those colleagues within this consortia can really play an integral role in providing subject matter expertise to think about what is the regulatory path that this particular drug might have to take to get regulatory approval. So for example, if you're working in a rare disease, how are you going to design your trial? Is it going to be your typical do 2 randomized controlled trials? Well, probably there's not enough patients in the world to do that, right? So those kind of concepts, we try to get our colleagues involved that know a lot about maybe your specific therapeutic area, and provide some guidance and learning for you to take in and help your project make better. So then specifically about the TRxA Bridge Awards. Bridge stands for Bridging Research and Innovation in Drug Development Grants. So for this year, we have some new modalities that we're including. So the indications are listed here on the left. So we are limiting our indications to 3 big buckets. One is rare and orphan diseases, the second is brain health, and the third is pediatric indications. And we picked those because we have a lot of strength within the Critical Path Institute in those 3 areas. And again, those are pretty big areas. So most likely your disease will fall into one of those. If you're not sure, feel free to reach out to us and we can have a conversation. But modality-wise, we've traditionally started with supporting small molecule projects. And then last year, we did a pilot program around protein-based therapeutics. So this year, both of those are, again, part of the scope. And then what's new this year is in vivo gene therapies. So let me talk to you a little bit more in detail about some of those modalities that are not small molecules, because we typically know what we mean by that. But what do we mean by protein-based therapeutics or in vivo gene therapies? Oops, sorry, I went too quick. Let me get back to that. First, let's talk about money. So let's talk about the award amounts. So we have 3 different stages that you can apply for most modalities. For stage 1, we're looking for projects that are early lead molecules, and we need to develop that into a late lead series. You can ask for up to $250,000 for that. Again, we can fund anywhere in the world, so just convert US dollars to your local currency, and you get a sense of how much we can support. Stage 2 is when you have identified a late molecule, late lead series, but you really need to hone in on what your clinical candidate is going to be. That will be funded for up to $500,000. And then stage 3 is you have a candidate, but you need to do those expensive IND enabling studies that are required by the federal agencies. And this can be funded for up to $1 million. So modalities wise, the stage 1 is open to all of those 3 modalities I talked to you about earlier. So small molecules, protein-based therapeutics, or in vivo gene therapies. And then stage 2 and 3 are only open to small molecules or protein-based therapeutics. The reason is in vivo gene therapies is new for us, so we're trying to limit the scope of that a little bit just to stage 1 proposals. And as we learn and bring on the right experts to be able to provide the best support as possible, we will expand that to stage 2 and stage 3 at a later date. So now let's talk a little bit more about protein-based therapeutics and what we mean by that. So examples could be peptides or proteins. I think most people on this call would know what we mean by that. But then also regular bivalent or trivalent antibodies, and also antibody-drug conjugates would be part of, would be in scope of this RFP. For in vivo gene therapies, there's some examples here where gene replacement is in scope. So for example, using the liver as a factory to produce an enzyme that's missing into a baby's body, that would definitely be considered in scope. In vivo gene editing as well as gene silencing. What's out of scope is ex vivo manipulation of, for example, cells and then transfusing them back into a patient. That's out of scope for now, just because the manufacturing issues are a little bit challenging, and we need to grow ourselves and get more understanding before we expand into cell therapies. But hopefully this gives you a sense of your approach would be considered eligible. So let's talk about eligibility criteria in a little bit more detail. So for small molecules, for a stage 1 project, of course, the top 2 or 3 topics are always going to be relevant no matter what you're applying for. So the indication needs to be for a rare and orphan disease, a pediatric indication, or for brain health. You need to have established what's called a compound progression pathway with clear criteria. That's pretty much a flow chart of which experiments you're doing in which order, and what are the criteria by which you consider the drug or the molecule to be successful. So you need to define it ahead of time. It also indicates how many assays you need to have in place, what the throughput of them needs to be able to handle. So it really informs a lot of your practical and tactical experiment design. And then you need to have completed the target product profile. We require that for all of our projects. And the project for a stage 1 small molecule proposal needs to be an early lead optimization. There needs to have been demonstrated that there's optimizable structure-activity relationship. So you need to have, a couple of analogs. I'd say at least 10 or so, that some show that you can manipulate the molecule, you can change some atoms around, and that the activity does not fall off a cliff, or that you only have one that works and nothing else works. That will be a red flag. So showing that there's, again, the ability to manipulate the molecule to make it more stable, more selective, etc. That's quite important. The in vitro assays that you need to go through this compound progression pathway need to be in place. And you need to have the appropriate throughput to handle the volume of new compounds that you need to be making. So whether it's you that's making new compounds or whether it's a CRO that's making them, let's say you get 20 new compounds shipped to you every week, can you handle that? Or can you only do like 2 a week? And well, that's going to be a problem from a logistical standpoint, right? So we want to look at what's the throughput of the assays that you have in place. In vivo models should be available, but there's no need to have animal data. Now, it's encouraged, of course, if you have some, you'll probably be a little bit more competitive, but there's no need to have shown animal data. And for in vivo animals being available, what we mean not necessarily is that it's in your lab, but that there are animal models with a collaborator or maybe a CRO again, or maybe it is in your lab, that's fine too. You don't need to do everything in-house. We're happy to support, projects with multiple collaborators at multiple institutions in multiple countries even. It just needs to exist so that you don't need to develop an animal model from scratch, because probably it's going to be a delaying factor in execution of the project. And then the final point is that intellectual property either needs to be in place or obtainable. So you don't need to have filed IP, but you do need to have the intent to file up IP. Again, composition of matter or other similarly strong types of IP are what we're looking at. So then let me go through the other stages and the other modalities in this detailed fashion. The top is kind of grayed out because it's the same for a lot of them. Again, indication needs to be in scope, compound progression pathway in place, and the target product profile needs to be completed. But when we're looking at stage 2 proposals for small molecules, here you really need to have a lead series in place with characterized in vitro pharmacology properties like cell-based activity and ADME, so absorption, distribution, metabolism, and excretion. You need to know those kind of things. Optimized candidates from ideally more than one series are available, and you have clearly defined what you need to fix. What's the remaining optimization call? There's usually something at this stage where like, it all looks good, but if only the oral bioavailability was better, or if only the selectivity was better. Like, what is that if only aspect that you need to improve? And then finally, at this stage, you really need to have shown target engagement in vivo. So again, target engagement here, we're not necessarily looking for efficacy. If you have it, great, but you need to have shown that in an in vivo model, your molecule manipulates the target. And then for stage 3, you need to have selected a clinical candidate. Again, you need to definitely have characterized ADME properties. You need to have shown efficacy in an in vivo model by that point in time. You need to have characterized some toxicology properties. You need to have a GMP scale-up and characterization plan in place. And you need to have defined a non-clinical formulation. So again, if you're not sure, like, I'm a stage 2, stage 3, or maybe I'm a stage 1, but maybe talk to us, we'll be happy to give you some guidance there. For protein-based therapeutics, again, the top 3 items are the same, but in here for stage 1 PBT, project again needs to be in lead optimization. In vitro assays need to be in place with the right throughput. Now, for protein-based therapeutics, immunogenicity is a big deal, right? So you need to have assessed the potential of your protein to be immunogenic, and you need to have identified a de-risking strategy. At this stage, for stage 1 PBT, you do need to have shown target engagement in vivo. For small molecules, we're a little bit more flexible because it's a little harder to get the right PK properties and so on dialed in. For protein-based therapeutics, for most types of proteins, we know pretty well what the PK looks like, whether it's a peptide or an antibody. So at this stage, we do ask for some in vivo demonstration that your target is being manipulated by your PBT. And then again, IP should be either in place or obtainable. For stage 2, you really need to have identified your single or maybe even more humanized, potentially, a PBT molecule. It should have been profiled enough so that the further optimization needs have been clearly defined. The strategy to reduce that immunogenicity needs to be in place. You really need to have shown therapeutic efficacy in an in vivo model. Manufacturing can be a bit of an issue. So small-scale manufacturing needs to be available in order to execute a project, but you also need to think about the future. So cGMP manufacturing should be feasible as well as scale up. You don't want to make 100 batches of 10 milligram to get to your right dose, right? That's just not feasible. So you really need to think about scale up at this point in the game. And then also the last point is important, the enabling technologies. If you need anything to deliver your protein-based therapeutic, or if you need any particular technologies to manufacture it, you need to make sure that's available and it's not blocked by any exclusive IP that somebody can, hawk for themselves. For stage 3, you need to have selected a clinical candidate, again, characterized ADME properties, shown efficacy in vivo, characterized toxicology properties at that point in time. You need to really have a GMP scale-up and characterization plan in place, and you need to have defined your non-clinical formulation. So then the in vivo gene therapies, only stage 1 this time, will probably expand in later years. But for this part, it's really important that the vector only targets somatic cells and avoids the germline. And so you really need to think about too, is there a clinical track record for the delivery platform that I'm using? So, AAV is used a lot, so that one would be fine, LNPs and so on. When it gets a little bit esoteric and it's very experimental, it's not going to be eligible. You need to have shown therapeutic efficacy in vivo. Again, immunogenicity can be a big deal, thinking of viral vectors in particular. So you need to have identified a de-risking strategy. Small-scale manufacturing should be available. Again, cGMP manufacturing and scale-up should be feasible. And similarly to the PBTs, IP needs to be in place or obtainable. And you need to have thought about what are enabling technologies I might need to deliver my gene, to manufacture enough of my viral vector or my LNPs, making sure that there's no exclusive IP that blocks it. If you can get a license to something that is broadly available, that's totally fine. But we need to make sure to not get painted into a corner that you cannot get out of. So I know this was a lot of detail. We have a guidance document online that lists all of this in excruciating detail even more. So feel free to look on our website for more information and materials in which you can read this back at your convenience. So let's talk a little bit about the process. So this is number one is where we're currently at. So there's a call for pre-proposals out. We have a Salesforce-based grants portal, and the deadline is Wednesday, March 19. It will be midnight of your local time zone in which your computer thinks it's at. So when you use VPNs, do make sure that you're in the right time zone. But here we're really looking for a high-level description, non-confidential summary about your project. So things like, what's your target? What are the properties of your lead molecule? What roughly do you need to do to get this to the next step? This is reviewed by an internal group within CPATH called the Programmatic Review Board. So us as the team, we're not on this board. There's other people within the organization. So we can be your advocate, your champion, your mentor, your guide, and we have other people review this, and they determine which projects advance in the process. So the second step is that when you get invited, we will go under confidentiality. Those requests will be sent out in the middle of May. The deadline is a month later. This is tight. CDAs should be able to be executed fairly quickly, but work with your grants and contracts office and your tech transfer office to make sure that this gets in place. And after that's in place, we have a secure repository, and then we ask you to upload any confidential information. So for example, a chemical structure, if you're using a small molecule, what's the full data package around the lead that you have, and so on. We will review this just to make sure there's no red flags there. Most projects advance through this step, no problem, though we have usually every year 1 or 2 that fall out. But then you get invited for a full proposal. So those invitations will go out early July, and the deadline will be middle of August. There's a little over 6 weeks time for this. Again, through this process, we are available as the TRxA team to help you, review some sections or answer questions that you may have. And really, you need to put together a solid plan. This will then be reviewed by an external scientific advisory committee, and they will score the proposals. So the list of the members is currently on our website. We will probably grow this committee a little bit over the next couple of months, but there are a wide variety of people, maybe retired pharma executives, venture capitalists, clinicians, chemists. A nice broad expertise is present on the panel. And then in addition, what we also do is for each of the proposals, we try to do outreach to another nonprofit organization that is specifically for your therapeutic area. So then those nonprofits, usually a scientific director or whatnot, will review the proposal as well. So in addition to our standing committee, we will have somebody who really understands the space in which you're working, the patient experience, as well as the technical expertise. So then those scores get forwarded back to our programmatic review board, and they make the final selection of funding. So all this takes, unfortunately, more time than we wish it would take, but we want to make sure that this is done very thoroughly so that it's very merit-based. And we hope to make announcements of who gets funded in October. So what do we support then when you get selected for funding? Of course, money. Very nice start. But also, as I mentioned earlier, we have monthly project team meetings with a bespoke team that we put together just for you, with composition of both us as the TRxA team, but also the larger CPATH organization. You get feedback and advice from a scientific advisory committee. Of course, you get the reviews of your proposal. So upon project selection, you get some input. We also ask for an interim report halfway through your grant, which is usually about 6 months in. And they can help, provide some advice, maybe provide a connection, give some feedback on like, well, you're doing this, but how about that? And then also at the end of the project, we try to have them review your progress, again, make available any connections they may have, warm introductions to potential investors, things like that. And then also throughout all of this, we provide connections to any services or expertise that you might need. Let's say you have a toxicology question that may not be really needed right now, but you need to start thinking about it. We can pull in a toxicologist and help you answer that question, right, as one of the examples. So let's talk about some FAQs. Of course, we'll have time for Q&A at the end as well, but these are often coming up as we talk about this program with investigators as well as tech transfer officials. So some things I've talked about already, but just to reiterate some of these concepts, we'll go through them one at a time. So who is eligible? So faculty at universities or nonprofit institutions anywhere around the world. Now, you do need to be faculty or something similar at your institution, just because trainees, for example, postdocs, they tend to move around a little bit, and that makes it a little bit tricky from a contracting perspective. So you do need to be faculty. Are there any institutional commitments required if funded? Yes. So we do expect to engage in a negotiation between TRxA and institution to negotiate a reasonable project agreement commensurate with the funding provided. We have a template actually of this agreement on our website, and also in the guidance document you can find on our website. There's a link to it. And we highly recommend that you actually share this document with your tech transfer office, maybe with your grants and contracts office, just to make sure that the terms are acceptable to them. We haven't run into any issues. The big picture is we would like to get a small portion of the proceeds to your institution as a result of commercialization, and that percentage depends on how much we've invested in the project. So it's 5% if it's $250,000 or less. It's 10% if it's $1 million or more, or 7.5% if it's something in between. And this is modeled after a lot of other nonprofit organizations and their agreements. So we've always been able to work out an agreement that everybody's happy with. So what we fund. So there's the question of we get some time. So do you have criteria for what qualifies for in vivo gene therapy beyond the examples you gave? So projects must use a delivery platform with a clinical track record. So there's some things that are excluded I wanted to highlight. So exosomes or extracellular vesicles are excluded as delivery vehicles. Again, there's a lot of promise there, but from a reproducibility issue, manufacturing issue, there's a little bit unknowns there that we want to wait for that to shake out a little bit before we start including that. Replication competent viral vectors like adenovirus, VSV, HSV, that's excluded as well. And then also targeting germline cells. Just we don't want to get into manipulating any of that. So how the funding works. So are indirect costs or overhead costs allowed? Yes, but IDCs, indirect costs, are limited to 10%. And then pass-through costs to service providers are not eligible for IDCs. And then people ask, well, does TRxA funding all come to my lab? Maybe, maybe not. The funds are really awarded to the project and not necessarily your lab. So part of the funds may, for example, go to the CROs or consultants. So it's really up to you to decide how can I best spend these dollars to move the project forward as effectively as possible. What are allowable costs? So costs associated with defined project task, like lab supplies, animals, salaries are fine, core services, and so on. What's not allowed are general lab expenses, travel, equipment, tuition, or IP prosecution and maintenance costs. Because we don't become an owner of any the IP, we leave that all with you. And general lab expenses, I had an experience once when I was at the university where somebody tried to buy new lab coats for the whole lab, on the grant that we gave them. That's really not the spirit of what we're trying to do here, right? This is for driving your project towards the clinic. So anything that's associated with doing that would be allowed. Do I need to worry about having other funding sources for my project? No. In fact, we really like to partner with other funders to speed up your project. Now, of course, that doesn't mean you can double dip. You can't ask money for the same body of work, but there's so much to do in drug discovery and development projects that if there's aspects you already have funded by the federal government, by another nonprofit, by your university, if we as TRxA can fund a complementary piece to then make a much stronger data package, that actually would play really well with our programmatic review board and also scientific advisory committee, because it means somebody else looked at it and deemed it worthy of investment. And how do you find the right CRO if you need one? Some people are like, I'm so lost, I don't even know where to start. So if you require advice, TRxA can help identify suitable CROs for some of the work. We have no geographic restrictions. Management of the contract will be through your institution, not by us, but we are happy to help you manage the work to be done. Look at the reports they send, help you identify what you need to ask them. So in that kind of how do I deal with it, we're happy to be a thought partner in that. People also always ask, do I need quotes for my pre-proposal? You do not, especially not for early stage work. And also TRxA can provide some ballpark numbers for standard drug discovery assays like selectivity panels or PK studies to help you build your budget. Now for later stage work, especially if you're asking for $1 million and 800,000 needs to go to a CRO for a non-human primate study or whatnot, that's where probably we will need a quote, but we will work with you at that point in time to kind of give you some guidance, what needs a quote and what's kind of standard. And we just know a ballpark number, and we're happy to use those numbers in your budgeting. Then people again ask, do I need to have IP filed on my compound before applying? So hopefully throughout this presentation it's become clear that there is no requirement that IP protection is in place, but rather that the composition of matter is eligible for protection. And we do expect IP to be filed, and also that it's going to be filed internationally. We run into this in the US a couple of times where tech transfer offices only file within the US. In pharma, it's really not a good strategy because you need to have at minimum, I would say, the US and Europe, if not some Asian countries as well, just to attract further follow-on investors to put money into the program. So how do you apply? Well, you need to start by creating an account in TRxA's Salesforce grant portal. There's links that can be found on the TRxA website, and the TRxA website is at c-path.org/program/TRxA. If you go to the CPath homepage, you can navigate to it, right? And there you can find a link to this grant portal and create your account. And again, the portal will automatically close at midnight in your local time zone, or at least the time zone your computer thinks it in. So make sure you start early. If there's any technical issues, if your account, if your password's not working or whatnot, you've met us as the team. It's 4 of us. We're based all in Mountain Time in the US. So if in your time zone, we may not be able to see your email and cry for help in time until the deadline's passed, right? Start early, start a couple of days before, if not weeks before, so that you save everybody a lot of stress. Then people ask, how many projects do you fund? So we anticipate funding between 5 and 7 awards this funding cycle. And that's for all modalities together. So there's no predetermined number of awards for each therapeutic area or each modality you want. And really, it all comes together in one big pot, so to speak, depending on scores and so on. And we fund the best projects, irrespective of therapeutic area and modality. Now, when you will enter into the Salesforce portal, you will need to make a selection where you apply for a small molecule or a protein-based therapeutic or a gene therapy project. Now, if you make a mistake there and you're like, oh crap, I have a small molecule project and I accidentally clicked on gene therapy, contact us and we'll see what we can do to get you directed to the right proposal. But if you're, not entirely sure, you have something that maybe fits the protein-based therapy or gene therapy, you can probably do either one. Again, at the end, it all flows back into one big group of proposals that we evaluate. The difference is there because we have slightly different questions that we ask depending on the modality that you use, because immunogenicity is, for example, not really relevant for a small molecule and vice versa. So what's the chance of success? So it will differ from cycle to cycle. For 2024, so last year, We had 2 rounds together. So we had one main RFP in the beginning of the year, like this one, that was for small molecules only. Then we had a pilot summertime protein-based therapeutics one. But those 2 together, we received a total of 100 pre-proposals. 26 of those were invited to a full proposal, so that's 26%. And then of those 26, we funded 7, so 27%. So hopefully that gives you a bit of a sense. Again, every year is a little bit different. We've expanded modalities, but we narrowed down therapeutic areas. More people know about us. So who knows? If I was a betting woman, I would say between 100 and 120 pre-proposals is something that we'll probably get in this year. But, you know, that's an N of 1 based on gut feeling, not on any data beyond what I'm showing you here. And then, of course, people will ask, well, can I get advice? I have questions. So we are available for questions via email at TRxA@cpath.org. Again, it's TRxA@c-path.org. Please start with email. We'll try to answer as many emails as we can. If absolutely need be, we can hop on a call. In previous RFPs, we offered consultations of meeting-based, but due to volume and, you know, just us being just 4 of us, we have to limit that a little bit and cannot just hop on a call with everybody. So if you have a question, start with email. And again, we promise if we can't figure it out over email, we will make the time to get on a call with you. So that finished this up. So the next steps, again, to remind you, create an account in Salesforce. On the grant portal, you can get there through our websites at cpath.org/program/TRxA. And submit your proposal by 19th of March, 2025. And please, please, please start early. And if you have questions, again, email us at TRxA@cpath.org. So I'll leave this up for a few more minutes, or seconds, however it works out, so that you can scribble down those email addresses and that website. But for now, I'd like to invite my colleagues, Michelle, Mark, and Kyla, to join us, and then we can go through some Q&A.
Thank you so much, Micah, for that excellent overview. We do have some questions to many have been answered online, but we have some that we've saved for live discussion. So first of all, would oligonucleotides, sorry, that
Good job, Michelle.
fall under your definition of a small molecule?
There would be, yes, there would be. Yes.
Excellent. And can you, this is what the questions are all scrolling. Can you elaborate on the indications you're looking for, that TRxA is looking for in support of the area of brain health?
Mark, you want to maybe take that one? I'm happy to as well.
Actually, I'm going to kick it to you, Micah, if you don't mind.
Okay, that's fine. We'll leave the chemistry questions for you. So brain health, we're thinking of anything that, well, maybe we should have called it brain disease, right? But it's neurodegeneration is a big focus within Critical Path Institute. So Alzheimer's, Parkinson's, but also things like ALS, stroke would be eligible as well. So things that affect the brain. So also psychiatric conditions like schizophrenia, psychosis, that would also be eligible. So hopefully that answers your question. If you have a specific indication in mind, again, feel free to email us and we'll give you specific feedback on your particular indication.
That was an excellent answer. It answered like 5 other questions.
Oh, good.
So thank you, Micah. Next up, do intrabodies count as protein therapy? It's a therapeutic antibody expressed in target cells via adeno-associated viruses at the DNA for the antibody.
Yeah, that's one of those tricky ones, right? It's in between a little bit. I would say either route would probably work, although using the AAV as a delivery vehicle, I would probably put it in the gene therapy category, just from the question and answer based perspective and what we ask in the pre-proposal, and immunogenicity, manufacturing, and so on. So it is expressing, you know, a protein by a gene therapy vector. So let's put it in the gene therapy category.
Okay, excellent. And this is also what, in terms of what's in scope or out of scope, what about replication deficient vectors based on the viral vectors that we listed in the slides?
That would be fine. Yeah, it's the reason I called out those replication competent ones, it starts to get, into, again, another realm of therapeutic approaches with different considerations and how do you control the replication and so on. But replication deficient ones like an adenovirus delivery a transgene would be totally fine.
Great. Next up, given the high cost for protein-based therapeutics manufacturing development activities that wouldn't be covered by our, by, for example, a $1 million award, how are the GMP scale-up and characterization plans evaluated during the review process?
I feel like I'm talking too much, but I'll just jump into Zoom and do. I would say that we're looking at, are you aware of everything you need to do? And then, so we actually have a question around this whole activity chart, so to speak, of all the things that you need to do. And then it's, okay, so now you have $1 million, and which pieces can you cover with the TRxA money? I think it will become fairly obvious as you go through the questions in the pre-proposal and also the full proposal, where we kind of separate those two things a little bit. We do ask about this full picture because getting confidence that you know what that whole activity profile looks like is important. But then we ask specifically, so, in the next year for this $1 million, what can you do? And that then comes down to the nuts and bolts of your specific scope of work under this award.
And I'll take this next question. I'm going to give you a break. The question is, what about repurposing drugs that are already in clinical trials for a different disease? Repurposing drugs currently are out of scope for TRxA, so that would not be eligible. It wouldn't be a good investment of your time to submit a proposal for that. Back to our therapeutic areas, we have a lot of questions about how strict they are, how broadly they can apply. We already talked about brain health. What about rare and orphan diseases? How do we define that?
I would say you can use either the US or the Europe definition. They differ a little bit. So we do ask for a few words around your therapeutic area. So in your answer, specify why you are determining that this is a rare and orphan disease. But yeah, there's predefined numbers on both on the US and the Europe side, of focusing on those two mostly because that's where most of our applications come from. Again, if you're elsewhere in the world, maybe look at those two. I think the US is less than 100,000 people with the disease. I forgot what the European definition is, but it's slightly different. Ultimately, again, if you're unsure, send us an email and we'll respond. But we are, in general, more open to the fuzzy areas than super strict, I would say.
Great. Another question on rare and orphan disease. What if the target for stage 1 funding is both for rare and orphan disease, but also applicable to more broadly to other diseases? If the program is focused on the eligibility indication, does that make it fundable?
Absolutely, yeah. And we actually, I think that's a great strategy as well, where your first indication that you maybe go to clinic with or that you want this money for is for this rare and orphan disease. But then if you know there is a broader disease that can be treated with this approach, I think it's really attractive from a commercial perspective. But that's, so you can hint at it in your write-up, but do try to not dilute your proposal too much with saying like, oh, this can be treated for, these 3 or 4 diseases, all completely wildly different. I think reviewers will kind of roll their eyes a little bit. It's an excellent approach for maybe federal basic science funding. For drug discovery, focus is king. So focus on your main priority disease area to, in this case, to make it eligible for TRxA. Talk about the other ones as kind of an add-on for it. This could be done later as well, but then don't spend too much time on it.
Great, thank you. Another question that's come up, is there a limit on how many applications an institution can submit? And the short and sweet answer to that is no. So go ahead and submit that. For IND enabling awards up to 1 million, for example, our stage 3 awards, do we ask for matching funds or a development plan for manufacturing development and GMP activities at a CDMO?
Mark, you want to take that one?
Yeah, there's no need for matching funds. and I think Micah already covered this, that in terms of quotes, I think for the big ticket items, we'd like to see quotes. You know, and obviously reaching an inflection point, you know, $1 million isn't going to take you all the way to the clinic. But you know, how far can you get, and does that reach a key inflection point? You know, does it move the needle on the project?
Thank you. I think another great question for you, Mark. We have some questions about for those who haven't been into our grants portal and had an opportunity yet to review the pre-proposal applications, what do they entail?
Yeah, so the application is designed, and if you pull the guidance document, the questions are there, and there is an example proposal. But it's designed to be fairly brief. You know, we don't intend you guys to spend, you know, days putting this together. But I think it's about 13 or 15 questions of what's your indication, and it's all non-confidential at this stage. What's your indication? What's your population? What's your target? What are you going to do in the year? And it's designed to be brief, just to give us an overview of whether to move forward.
Great. Thank you. And I think this is another great question for you, Mark. How about repositioning of existing molecules, but chemically modified to increase efficacy? Would that technically be considered a new chemical entity?
I think if it's outside of obviously you'd know the patent literature around that chemical entity. If it lies outside that patent, then, you know, by definition, I think it's a new chemical entity. If you're improving on something that's non-obvious and you can get IP on that material, I think it would be defined as an NCE.
Okay, great. And I don't think I've verbalized this question yet. If I have, Micah, forgive me. I have a project that uses a phenotypic assay, but we do not know the target. Would this be eligible for funding?
That's a good one for Mark too. I'm happy
Okay.
to.
Yeah. It would be eligible for funding. I think it's probably going to be, from experience, one that's maybe not as competitive. But I think you know, as long as you can demonstrate your phenotypic assay is you know, it's robust enough that you can run a medicinal chemistry campaign through the assay, and that, you know, maybe that there's efforts in place to pull down the target. I think that would be a stronger application.
Yeah, and to add to that, we actually are funding currently an app project based on a phenotypic screen. They have to try twice, so and it also illustrates that you can resubmit and learn from the feedback and so on. But yeah, I would agree. It's doable. You have a little bit higher bar to clear probably, but if you have already efforts in place to do the target identification, then it should be possible.
Great. This is a great question for you, Mark. What about, can we talk a little bit more about target product profiles, right? For faculty who don't know or understand what it is, what kind of guidance can we provide to our applicants through this?
Yeah, I think
process?
I hope I'm not going to misspeak, but I think there is a target product profile example on our website. But if you, if you Google target product profiles, there's a lot of papers. Paul Tebbe is an author who wrote a good paper, TEBBE. But it would just describe how one would go about putting it together. You know, there's templates available through NIH and FDA and these kind of places. So I would just essentially download one. And it really, you know, as you put it together, it's going to, you know, based on your knowledge of the therapeutic, and it varies, you know, if there's no drugs, you know, the therapeutic, you might be able to deliver it non-orally because it's tolerated by the patient population. But there's a lot of good literature out there and a lot of good examples. And then, you know, as you go through and build it, I think you'll be able to see what's really where you're aiming for your project.
Great, and I think we have time for one more question. If there are some competing drug programs for my project, perhaps not against the same target, will we require head-to-head animal data or additive effect data to help with justifying the proposal?
Well, let me take that one. It depends. I would say I think the approach that's being described in the question is a good one. Ultimately, you need to convince an experienced panel of drug hunters that it makes sense to invest in your project. So if you are clearly superior to existing standard of care, and you showed it head-to-head in an animal model, you have an obvious advantage, right? If it's more hypothetical, we've had some examples of that, I think, go through our process. There's going to be a little bit more scrutiny going on. But acknowledging that those exist and highlighting the differences is key. We've had a couple of experiences where people either didn't realize there was a competing program, same target or different target, doesn't really matter, but that there was something in the pipeline for that particular patient population that would improve their condition. And our reviewers found out about it or knew about it, and the applicant did not mention it. That's not good grantsmanship. So like with anything, tell your story, be upfront about potential, things that may be a mark against you or whatnot, but then, set it up as a straw man so that you can address that issue and make the case why your project would be a really good investment. So there's some grantsmanship storytelling that comes into play there, but there's not a requirement for head-to-head comparison to be eligible, but I think from a competitive standpoint, it would be good form to do so. Now, there's also a little bit of a difference between the pre-proposal and the full proposal. So pre-proposal is going to be only non-confidential information, right? So you may not even have that, you don't want to share that kind of data unless it's published in a paper. In the full proposal, which is under confidentiality, our scientific advisors are also under full confidentiality with us, and then also with your proposal. That's where you can be a little bit more strategic into what experiments are showing off your new therapeutic in the best light possible to make the reviewers excited about it.
Great. I'm going to quickly answer one last question. We've heard news, late-breaking news about federal grant funding interruptions, right? A lot has been happening in DC. Will this impact TRxA's funding cycle? And the answer to that is no, right? We are not federally funded, so our RFP will continue uninterrupted.
Yep.
Okay, that takes us to the top of the hour. And we are so grateful for everybody who joined us and all of the great questions. Again, if you excavate any questions after this webinar, please feel free to email us at TRxA@c-path.org. Thank you, everyone.
Thank you. Have a great day.
Bye-bye.
Bye.
Thank you.